Hello and welcome to Nikolai's genetics lessons, and today's video going to be conceptual for those people who study plant breeding and selection, not necessarily plants, but it also can be animals. So here's a problem. Two inbred strains of corn were crossed to produce an F1 generation, which was then intercrossed to produce F2 generation. Data on ear length from sample F1 and F2 individuals gave phenotypic variance of 15.2 centimeters and 27.6 centimeters respectively. Why was the phenotypic variance greater for F2 than for F1? So take a look at this picture. This is corn ears. This is exactly where kernels are, edible part of the corn. We are told that we took two inbred lines. What does it mean? From genetic point of view, that means that for every locus, this plant would have the same alleles. So, for example, for the gene A, plant would have two dominant alleles. For the gene B, say two recessive alleles. And for the gene C, two dominant alleles, and so on. So would be homozygous at each locus for each allelic pair. Now let's say for the gene D, this plant is going to be homozygous recessive. And now we cross with another parent. Another parent also is going to be a representative of the inbred but different line. So genotype would be, say, homozygous recessive for the gene A, homozygous dominant for the gene B, homozygous recessive for the gene C, and homozygous recessive for the gene D. And as you see, it is not necessary that two inbred lines would be opposite of each other in respect of certain loci. For example, locus D for the gene D can be homozygous recessive in both lines. But we also expect that for certain loci, these two strains would be different. For example, one would be homozygous dominant, another homozygous recessive. So this is example of two inbred lines. And let's say we took one, which is parent 1, and second, which is parent 2. But anyway, we can take any plant from this inbred line, and all would have the same genotype, because this is inbred. So they don't have genetic variation. And this parent also can be any plant, because if we take any plant from this inbred plants, this plant is going to have the same genotype as other representative of the same inbred line. And what's going to happen in F1 generation? So here is F1 generation, and this is parental generation. F1 generation is going to be hybrids. So for the gene A, this generation is going to inherit from parent 1 dominant allele, from the parent 2 recessive allele A. For the gene B, would inherit dominant allele from parent 2 and recessive allele from the parent 1. For the gene C, is going to inherit dominant allele C and recessive allele C from the parent 2. And for the allele, for the gene D, would inherit two recessive alleles from both parents. Now, if we cross this representative of the F1 generation with itself, that means with the same genotype, because all F1 generation is going to be genetically similar, genetically identical. In the next generation, we are of course going to see different genotypes. For example, plant can inherit dominant allele from parent 1 and dominant allele from parent 2. So can be capital A, can also inherit dominant allele from parent 1 and dominant allele from parent 2. So can be capital B, can inherit recessive allele from parent 1 and recessive allele from parent 2. So can be small c, and of course would be small d. Another variant can be capital A, small b, capital C, small d, and many other variants are possible. For example, small a, capital B, capital C, small d, and many other different variants. That's why in F1 generation we are not going to see much variation. All variation that we can see would be environmental variation. Of course, if genotype of the F1 generation is going to be the same, it doesn't mean that all ears are going to be of the same size, because all plants in a field would be affected by different environmental factors, such as, for example, would get different amount of water, of the sun. Some can be damaged by the insects. There also can be competition in the root system and many other factors like soil composition and so on. So we expect that in hybrids, which is F1 generation, some variation, but all this variation is going to be due to environmental factors. So in F1 generation, variance phenotypic would equal to variance environmental. And as for the F2 generation, here we would see that variance phenotypic would equal to variance environmental plus variance genetic. One more time, in parental generation we have two inbred lines. So parent 1 have genetic variation 0. Parent 2 has genetic variation 0. Just like in F1 generation, genetic variation is 0. But in this inbred line, and in the progeny, we would see some variance, phenotypic variance, but this is only going to be due to environment. But in F2 generation, we would see different genotypes, and phenotypic variance would be due to environmental variance and genetic variance. Another example, imagine that this line represents variance in size in F1 generation, and this one would represent variance in size of the ears in F2 generation. So you see that here the variance is going to be greater than in the example with F1 generation, or if we also take inbred parental generations from both sides. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day, and see you in the next video. Goodbye.